So good morning, everybody. I would like to thank the organizing for this opportunity to talk about this project that we did at INRAE on the estimation of plant area index and the distribution of plant area index using LiDAR on terrestrial, using terrestrial LiDAR. So, well, most of us, we know what a LiDAR is. So it's essentially an active sensor that measures distances. So a light is emitted in a given direction, is reflected back from the canopy, and from the difference in time of phase, we can determine the distance between the LiDAR and the impact. So when you do that a large number of times, what we get is a 3D point cloud of our canopy. In species or architectures where are relatively simple, just we can estimate leaf or even the plant area by reconstructing a mesh directly from the point cloud. However, in complex and dense canopies, as with canopies, probably the reconstruction of a mesh surface is not the most adapted approach. So the objectives of this study was first to develop a method based on LiDAR to estimate the distribution of plant area in wet canopies in the field from LiDAR point clouds. The second was validating that approach with destructive field measurements. And finally, understanding the role of different factors, like, for example, the LiDAR viewing geometry, or also the possible differences in the architecture of the genotypes that could eventually impact the plant area index estimations. So this work relies on 3 field trials that were conducted in 2022 and 2023 in 2 phenotyping platforms in the south of France. So we have always the same 10 genotypes, breed with genotypes, shown with different sowing dates, water treatments, and sowing densities. And we conducted a total of almost 300 destructive measurements of plant area index in the microplots. These are the marks that you see at different growing stages that goes from the end of tillering until anthesis. So in these phenotyping platforms, we have the Phenomobile ground robots, and it's with that we acquired the LiDAR point clouds. So basically, the Phenomobile has a system of 3 LiDARs. 2 of them are looking downwards, nadir, and a third one is looking at 45 degrees. So there is a large overlap between the footprint of the third LiDAR. Basically, they operate like a patchwork sensors. So the LiDARs scan in the direction perpendicular to the rows, and the measurement here moves in the direction parallel to the rows to generate a point clouds of about 30 million points for a microplot of 10 square meters. So here you see the area that we destructively sampled. So how did we estimate the plant area index? So first, we have the original LiDAR point clouds, and first we estimate the position of the ground. So we analyze the impacts, the frequency of the impacts at the bottom of the canopy. We fit a linear model with a small, offset to prevent the effects of soil roughness in the results. And then we voxelize the point clouds. So we divide the scene in voxels. The voxel size in the X and Z direction is quite low, and we define relative to the row distance, so it's 1/10 of the row distance. And in the direction parallel to the rows, the voxels are a little bit longer to maximize the number of beams that are incident in the voxel. Then what we do is to compute for every voxel the gap fraction. So it's the probability that a beam passes through the voxel without impacting in the canopy. For that, we can trace the trajectory of every beam. So for every voxel, we count the number of voxels that goes through, and we divide by the number of incident voxels. And like that, we have this gap fraction probability. At the end, what we have is a 3D map like that. For every LiDAR, we have for every given voxel what is the gap fraction on that volume. Then, from this gap fraction, we calculate the plant area index based on the Beer-Lambert law. So Beer-Lambert law is a kind of physical optical model that is used in many instruments that we use in the field to determine leaf area index, plant area index from optical measurements. From septometers, digital hemispherical photographs, LI2000, they rely on the Beer-Lambert law to estimate leaf area index. The problem with Beer-Lambert law is that you have two unknowns. You have the plant area index, but you have also leaf inclination. You need, when you have only one observation of the gap fraction, you need to do some hypothesis on how the leaves are inclined in order to derive plant area index. The positive point with the LiDAR system is that we have the same voxel observed by 3 LiDARs at the same time. So at the end, what we have is a vector of 3 observations to solve a problem of 2 unknowns. So at the voxel level, thanks to these 3 observations, we can numerically invert the Beer-Lambert law to estimate at the same time plant area index and average inclination angle. So how do we do? In order to be robust, we follow a slightly different strategy. What we do is to consider that all the voxels at a given height have the same inclination angle, but that PAI changes from voxel to voxel. The strategy is to sample for every set position. We sample the inclination angle from 0 to 90 degrees. For a given inclination angle, the PAI can be solved numerically by solving this linear model. So what we have just to do is to select the average inclination angle that produces the best fit between the observed and the model data. This is a little bit technical. I'm not going too much in detail, but if you are interested more specifically in some questions, please ask me later. And then from that inversion, what we get is a 3-dimensional map of the distribution of the plant area index. And of course, the vertical distribution of the leaf inclination angle. We test, based on this approach, we test different hypotheses. We want to know first if the third LiDAR at 45 degrees contributes, at which point contributes, because most of the optical systems have essentially nadir looking sensors. We want to know what is the contribution of the third LiDAR. And also we test several hypotheses on the average inclination angle. The first is that, okay, we don't know, we don't have any prior information of average leaf angle, and then the inversion of Beer-Lambert law will estimate both PAI and ALA, unconstrained. The second is that, okay, we introduce a penalty term in the inversion to avoid that the average leaf angle takes values that are unrealistic. And the third one is that we do, as everybody, in most of the cases for field crops, they fix the average inclination angle to 60 degrees, which is a spherical model considered as reference in most of the optical sensors. Okay, and then of course when we from these 2D graphs, from this 2D distribution of PAI, what we do is integrating PAI at the canopy scale only from the voxels that overlap the area, the exact area that we have destructively sampled in the field, and we compare canopy line measured with canopy line derived from these maps. So the results indicate that the hypotheses on the average inclination angle are extremely important. So when we leave the average inclination angle to vary freely, we have a massive overestimation of the plant area index. So there is a need, I will explain you later why. We have to constrain average inclination angle in order to get satisfactory results, either by inserting the penalty term that I explained before, either by fixing the average inclination angle to 60 degrees. And in that case, in both cases, more or less, it will work fine. The results are slightly better when we use the 3 LiDAR instead of the 2 LiDAR, but more or less the overall root mean square error is okay, is satisfactory. Why do we have this massive overestimation when we leave the inclination angle free? Well, these are the estimation of the average leaf angle with this configuration and the hypothesis of ALA free for every genotype in the 2 sites. And this is when we do that, when we constrain with a penalty factor the average inclination angle. What we see is that in this case, we have the bias in this in the ALA. This massive overestimation is due to unrealistically high estimations of the average leaf angle, which are due to a slight bias in the gap fraction due to the LiDAR spot size. If I have time, I could explain that later. What we do with the penalty term is constraining the average leaf angle to vary around 60 degrees, depending on the genotypes, which is the expected value that we know a priori should have a crop like wheat. So this, how to determine the parameters of this penalty term. So what we have here is the variability of the root mean square error in the 3 and the 2 LiDAR configurations, depending on the 2 values of this penalty term. The crosses indicates the hypothesis where the ALA is fixed and the ALA is constrained. So basically there is a big area in the space of parameters where you can adapt the values of both parameters, but basically the root mean square error is the same. We can adapt these parameters to let the average leaf angle vary across genotypes, but basically we have more or less the same error as if we fix the average inclination angle to 60 degrees. So can we just keep average leaf angle constant and we forget about everything? Well, the result says that this is not exactly a good idea. So these are the correlation between estimated and destructive canopy leaf area canopy plant area index by cultivar. So when we fix the average leaf angle to 60 degrees, especially in the 2 LiDAR configuration, we have relations that are specific for the cultivar. And this is obviously not what we want from a high throughput phenotyping perspective. So the 3rd LiDAR helps a little bit to mitigate, but however, these differences appear. However, if we leave average leaf angle to vary with the penalty term, what we have is that almost all except these 2 varieties, let's say the relationship between observed and estimated plant area index seems to be independent from the cultivar. The hypothesis is, okay, when we allow them to vary, we are dealing with possible differences in leaf inclination across the cultivars. If we keep the ALA fixed, these differences in architecture will impact the plant area index. So actually, with that approach, we see that in the 2 sites, Toulouse and Moggio, there is a kind of classification between in the inclination of the cultivars. We have some cultivars that are a little bit more erectophile and some of them more planophile. Are these estimations realistic? Well, if we look at the distribution of plant area index, we have this orange curve, which is the vertical variability of the inclination angle. And these are photographs, RGB photographs that are taken simultaneously to the point cloud, because also the measurement head is equipped with an RGB camera. What we can see is, for instance, this Sacramento cultivar. We see clearly that has a very erectophile habit, and the estimation of the ALA says that we are about 70 degrees inclination in the upper side of the canopy. Chevignon has a kind of intermediate average leaf angle, so it's more or less compared to Sacramento what we observe here. Renan is a kind of outlier because it's a cultivar with very large and very curved leaves that decreases a lot the average leaf angle, especially at the top of the canopy where the curvature of the leaves makes the average leaf angle decreases. And also, due to that particular architecture, this distribution of the leaf inclination, we don't appreciate clearly the raw structure in the plant area index. And then our grain has a particularity that the flag leaves is almost planophile. So this we can see here, this short decrease in the average leaf angle at this point, which indicates more or less the insertion height of the flag leaf. This increase below of the average leaf angle that comes with a decrease of the plant area index would indicate apparently the midpoint between the last two internodes, where normally you have, theoretically, you would expect that you have less leaf area if the flag leaf is flat. So, okay, this suggests that the estimated average leaf angle that we produce both across genotypes in the vertical profile makes sense. So, in relative terms, I would say that these estimations of the average leaf angle describes what we have in reality, although we cannot know the absolute accuracy of that estimation because measuring the leaf inclination manually in the field is extremely challenged. So we cannot test the absolute accuracy. So just to finish some conclusions, so we proposed a model for dense canopies that allows to describe the variability in 3D of the plant area index and the average inclination angle. So the validation of the canopy shows satisfactory results, but however, we need to have some previous knowledge on the average inclination angle to have these results. The best strategy is constraining the leaf angle with a penalty term. However, this penalty term is a priori instrument dependent, and this is a bit of an issue. I mean, you need to change it probably from time to time. We have seen that the space of parameters that provides a satisfactory root mean square error is large, but anyway is dependent of the instrument. And last, that we cannot validate in absolute terms the average leaf angle that we estimate. We cannot validate it in absolute terms, but at least in relative terms, the variability that it describes makes sense according to the evidence that we have. Finally, the future challenge is trying to use this data with other phenotyping data that we collect from Phenomobile to estimate, probably using an intermediate architectural model, more detailed organ level traits. For instance, the individual leaf area per strata. And just a last word to say thanks to all the colleagues from the FAST project in France that helped a lot in this massive validation with field data that we have conducted in 2022 and 2023. Thank you very much.
Okay, thank you very much for your talk. Very interesting. I was curious, can you share a little bit more about the canopy itself is very complex and dense, and yet your voxel size you made quite large. What if you would reduce that voxel size? What would be the impact on your approach?
Actually, the voxel size is kind of optimized in order. I mean, it's important that in the X and Y, X and Z directions, in the direction perpendicular to the row and the canopy height, to have a voxel size small enough to avoid that you have clumping of leaves at the voxel scale. Because when you have clumping inside your voxel, leaf clumping inside your voxel, the Beer-Lambert law doesn't apply well. as we assume that in the direction parallel to the rows, the distribution of leaf area should be more or less homogeneous because you don't have a row structure, we can increase the size of the voxel without violating the assumptions of the target medium. Could we reduce the voxel size in that direction? Yes. The problem is that when you reduce the voxel size, the number of incident beams is lower, and then the computation of the gap fraction, especially as you go in depth in the canopy, becomes more and more robust. So either you average your gap fraction in the y direction, either you enlarge the voxel size in that direction in order to have satisfactory results. So more or less, that reaches the same point.
Hey. Yeah. Thanks for your talk. I have two small questions. First is I wonder how the wind affects the LiDAR measurements, because from my experience of working inside a wheat canopy, even a slight breeze moves the leaves a lot, and even the whole plant. And in a second question would be if you compare the LAI results with like septometer measurements or SunScan measurements, something like that.
For the first question, it's a very interesting question. Actually, we try to avoid acquisitions on wind first because, let's say, that in the LiDAR acquisition, if the leaf moves, you can have the same leaf falling in different voxels, and also, let's say, the co-registration between the voxels number 1 and number 2 and number 3, which is a little bit backward, becomes a mess. Apart from that, the fact that you have a preferential orientation of leaves when there is wind also, in theory, violates the target medium assumption of the Beer-Lambert law. So, yes, when there is a lot of wind, it's preferable not to go with the Phenomobile. I think with any optical method. The second question is no, we didn't compare with other optical methods. I mean, when we wanted to validate, we prefer to validate that with destructive data, also because canopy-level methods as septometers or LAE 2000, etc., they estimate what is often called an effective leaf area index. I mean, you are including the effect of leaf clamping, and your estimated leaf area index or plant area index may not be exactly similar as the actual one from a destructive measurements.
Thank you. Interesting presentation. So it's nice to see that with 2 LiDARs we can do as for 3, because we went for 2. So it's nice to see these results. I have 2 small questions. The first one is, did you do the same for other leaves throughout the cycle? I mean, F-1, -2, and to see the angle too, and see how it is, especially, I know that the flag leaf is the most important one when we talk about, seed production, but what about the other leaves and the behavior? And did you also do any kind of correlation between the seed yield and especially the flag leaf angle, or what you did with the flag leaf, and the number of tillers per plant?
And the number of?
Tillers.
Okay. No, we are, at this moment, we are based, for your first question, we have a Phenomobile acquisition every 100-degree days. So let's say we cover the whole cycle, but we still didn't analyze. We analyzed the variability of plant area index at the canopy level, but we have not yet started to analyze specific traits on different canopy depths. We have, we can discuss about it later. We have a project that hopefully will start next year to use a 3D model to transform this two-dimensional description of the plant area index per voxel into a kind of more specific traits like the average individual leaf area per order in the main stem. So that would be the idea. And we didn't have evaluated the relationship between not yet the relationship between the area of the flag leaf with the kernel number, not.
Not only the area, but the angle too.
Okay.
So not only the area, it's the angle too, because there is good correlation when it comes to the flag leaf and CDL, because this is the leaf that contributes the most to the CDL. Okay. But I don't know if it's always interesting to see such results, but what about the correlation with the CDL in terms of differences here? It's some kind of...
It's definitively very interesting. I mean, so I think we can discuss it later. We didn't thought about it, but it could be a nice topic to explore, because yes, depending on the normal between the leaf and the sun position, exactly we may have differences on the total absorbed light. And yeah, thank you very much.